Hello, ladies and gentlemen. My name is Terrnika Jackson. I am a senior at school at Walls High School, and the title of my project is Genetic Variation versus Environmental Contaminants in Striped Bass in the Anacostia River. So my hypotheses are as follows. My null hypothesis is that there is no correlation between striped bass genetic variation and endocrine disrupting chemicals, also known as EDCs. And my alternative hypothesis is that there is a correlation between striped bass genetic variation and EDCs. So before I go into my experimental design, I just want to give you a little bit of background information on my project. So I interned at Georgetown University with my mentor, Matthew Hamilton, who is a population geneticist as well as a professor at Georgetown University. And he really helped me kind of hammer out the details of my project. And the whole point of my project was to see if there is a correlation between chemicals that are found in the water and if they are in the Anacostia River specifically, and also if these chemicals were mutating a specific gene in fish that were responsible for estrogen production. And the reason that I really looked at this specific gene was because of Dr. Vicki Blazer, who I had the chance to interview last fall about her studies and what she thought about intersex being so prevalent on the east coast of the U.S. So the way that I did this was with polymerase chain reaction, or PCR, primer design, and the gene aromatase B in striped bass. So striped bass was not my first choice in a model for a fish. I really wanted to work with smallmouth bass because that's what Dr. Vicki Blazer herself worked with, and I wanted to see if I could kind of challenge her results in her studies. And obviously not on the same level, but I definitely wanted to just kind of give her food for thought. So polymerase chain reaction was my big step in my project. So what I wanted to do was I wanted to find the gene that I thought that would have been mutated by the endocrine disrupting chemicals. And endocrine disrupting chemicals are just chemicals that it's not like one specific chemical. It's any chemical that has the capacity to disrupt the chemical signal of the endocrine system, which is the system that controls hormones, and which including sex hormones like estrogen and testosterone. And so PCR is a tool that is used to amplify millions of copies of a specific part of the DNA. In my case, aromatase B, the gene that I'm looking at, and you're able to amplify it. And the reason I was wanting to amplify it was to get enough DNA to sequence. And the way that I did PCR was through primer design, which are like tags, and a long, well, let's say that you have a huge text and you want to just look for a very specific part of it. It's really just like searching Control F on a PC. It's really looking, it's sending in specific nucleotides of DNA to look for a region of the genome and amplifying just that part. And it's really cool, and it was really exciting to learn, and it took me a long time because striped bass have not been sequenced before, and the way that primer design usually works is that you have a template sequence that you're working with, and I didn't have that. But I was able to make a successful primer, and that was really exciting for me. So my findings, even though I had a successful primer, my primer designs were inconclusive. I did 3 primer designs, or not primer designs, I did 3 PCRs, and the way that you check your results through PCR is something called a gel electrophoresis, where you run your results through an electrical current, and it shows the different DNA pieces that have been amplified. And it was successful in my second try, not very successful in my first, and not very successful in my third. So my mentor explained to me that PCR is something that's really hit or miss, especially with the primer design. And sometimes it doesn't work and sometimes it doesn't. And I do vividly remember when I first started, my teacher told me that primer design is a little bit of witchcraft and a little bit of science. And now I really know what she's talking about. But the good thing is aromatase B was amplified. And I do know that because of my 12 out of the 18 samples that I did were that I was privy to actually did have amplification. I wanted to sequence this DNA to see if there were any single nucleotide differences, but I did not. Well, I was not successful with my design, so I wasn't able to. So my limitations, I had a lot of limitations, but I think the biggest one was that my fish weren't actually from the Anacostia River, and that was kind of misleading. Well, I don't want to say misleading. My fish samples were from the Paxton River in Maryland, and the Anacostia in the Paxton River is part of the Chesapeake Bay. So I wanted to. Well, I thought it would be okay to use that, even though it wasn't specifically from the Anacostia. I really couldn't get fish. They weren't accessible because I was doing scientific research, and a lot of organizations don't give fish samples for scientific research. Also, I wanted to correlate chemical concentrations of the chemicals to genetic variation. Like the more varied a fish gene was, the more chemical concentration I had. That's the kind of thought I had, but it's very expensive to do chemical testing, and my mentor and his organization did not have the money. And also, I had just under a year to do this, which I thought was a lot of time, but it really was not at all. If I had the chance to expand my research, I would want to test more genes for variation, not just aromatase. I would try and do a little more research on other genes that could be susceptible to mutation due to chemical um, concentrations or chemical exposure. Also, I would do it in more fish. Striped bass is a really good fish. It's a really good model fish, and a lot of fish are already model fish, like the zebrafish, tilapia, goldfish, and more. And I really wanted to see if I experiment with those as well, see if there's any patterns across for interspecies and stuff like that for fish. And also, I will actually want to go through with my chemical testing for a stronger correlation. Thank you so much. I hope that my research interested you, and I hope that you were impressed. I worked pretty hard on it, and I hope that you guys can see that. Thank you so much.